Voice of the Sea, learning from experts across the ocean. Welcome to Voice of the Sea. In this episode, we're building supercorals with biologist Ruth Gates at the Gump Research Station in French Polynesia. Ruth and her research team are conducting experiments to learn what makes some corals able to withstand extreme temperature fluctuations and how this information can help biologists prepare coral reefs for our changing climate. With rising global temperatures and increasing ocean acidification, coral reef resiliency is critically important to food sustainability and to protection of our coastal communities. We met Dr. Gates in the lab to learn how she identifies these so-called supercorals.
This is our molecular lab, and this particular institute, like the Hawaii Institute of Marine Biology, has extraordinary indoor lab facilities, and that's actually for a field station is relatively unusual.
Why is it important to have an indoor facility? I mean, I understand why you'd want to do research here and you have great access to the outdoor marine environment.
Having these labs so close to the coral reef actually allows us to bring samples in, and we can actually look at them very closely under a microscope. So we have a variety of different microscopes that allow us to look at the living sample. We can then sample a particular location on that coral, and we can actually do the analyses here on site. Why that's important is because if we have to resample those corals, because perhaps something goes wrong with the analysis, we can immediately go out and do that. We can bring corals in and we can put them back on the reef having done things, and we can tag them. We're able to manipulate them, take them back out, tag them, and then we can look at them the next trip that we come down. As we look in a lab like this is sort of it's the LTER lab, the Long-Term Ecological Research Lab, and you can see behind us there's a fair amount of empty space, and this is where our students sit. So you have to have somewhere to work. When we come over to this bench here, this bench is really a bench where we do a lot of physiology, and what we mean by that is we're measuring how much oxygen a particular coral breathes. It seems bizarre to think that things are in water, but that they're breathing oxygen. But of course, like all living organisms, they breathe the dissolved oxygen. They use it and they produce it. Same with the carbon dioxide in the water. They use it and they produce it. What we're very interested in is the way in which processes that are originating in things that you can't see are actually driving patterns on the reef. And so when you swim out on the reef, you see, for example, a reef that's dominated by corals, and they're all different colors. And you know, some of the questions that we're interested in is, why are they all different colors? And that's not something that you can really study just by swimming around. You can't know why they're different colors.
The University of Hawaii's Sea Grant College program focused on Hawaii's coasts and its communities through sustainable development, safe seafood supply, sustainable coastal tourism, hazard resilience, and healthy coastal ecosystems. Hawaii's Sea Grant.
You're watching Voice of the Sea. Welcome back. We're learning how some corals are super athletes and how populations of these corals may help to save reefs with Dr. Ruth Gates.
Why does a particular coral withstand environmental disturbance? So my lab particularly is interested in why some corals survive conditions that kill others. And we look at things like their genetics. We look at who they associate with. So corals are really remarkable organisms because they are consortia organisms. They rely on other things that live inside them to survive. Those other things are small plants that live inside them. They're like factories that generate food for the coral. The coral eats that, and the coral is dependent on them. They can associate with different types of algae. We want to know why associating with a particular type or another type gives you either an advantage or not. in terms of withstanding change. What we would do is we'd have a tiny, basically a small dish with a tiny piece of coral in seawater. We'd bring it over to the lab bench, punch a tiny biopsy, much like you were biopsying a mole, and we would take that with a sterile punch straight into a sterile tube. That tube contains sterile liquid. That liquid goes through a series of processes that allows us to extract the DNA from it. And that DNA looks, you can't actually see it, it's just a small pellet of material that we dissolve back into sterile liquid. And then we could subsample that liquid and do any number of tests. We can find out who that person is, we can find out who they associate with, and we can actually examine various parts of their function by looking at particular genes that control how regularly they reproduce or how quickly they will grow. So we can do a lot of different types of tests with a relatively small sample. As the coral respires or breathes, it will produce or consume oxygen. So the oxygen levels will go up or down, and that will depend on whether we're in the light or the dark, actually, because there's another process that corals can do, which is something we can't do. That is photosynthesis, and that is the fixation of carbon dioxide to create food.
And does the coral do that itself?
Well, it depends on your opinion. So the coral is a single organism that has other organisms living inside it, and it has these tiny unicellular plants that live inside its own cells, which is crazy. Corals, over many, many millions of years, have allowed this plant to first enter, and then it's essentially farms it within its tissues. So in a particular cell layer of the coral, you'll find these plants in every single cell. And those plants live there. They get protected from the outside by being there. And while they're there, they do what all plants do, is they are able to convert inorganic or gaseous carbon dioxide into organic food stuff. So stuff that you would want to eat, right? We can't, when we breathe carbon dioxide, we can't use it for food, but plants have done this amazing thing where they take that what we breathe, they unite it with water, essentially using sun's energy, and from that they can create a sugar or a food that you would want to eat. And that is actually the base of the food chain on the planet, which is why it's critical that we don't take all the plants off the planet and we don't cut down all our forests, because that is so fundamental to our food supply. So corals do that, and when they do that in the chamber, because CO2 is being used and water is being absorbed, what gets produced is a sugar plus oxygen. So they breathe oxygen, but they can also produce oxygen, but only in the light when photosynthesis is happening. So things happen differently in the day and the night for corals, and the amount of oxygen in the water will change and reflect the combination of those two processes during the day, but only breathing at night. But what's interesting is if you put two corals side by side and you do exactly the same thing, you may find that one of them breathes more than the other, or one of them is able to do carbon fixation and photosynthesis at a greater rate than the other. And this is often an indication of a genetic difference between the two, or maybe it's something to do with them being at different stages of development. Much like children, they have very high metabolic rates, you know, they're constantly running all over the place, but, you know, by the time you get to my age, you kind of just want to sit in a chair. And the amount of oxygen I produce or use compared to the amount of oxygen a child is a totally different ball game. So we see those types of phenomena in our examination of the oxygen use in these chambers. One other way we can do that is this is where we can examine the same coral from two different locations. So we might do a series of experiments here in Moorea with a coral species that also occurs in Hawaii. And when we go to Hawaii, we have exactly the same setup, and we will do exactly the same experiment with our corals in Hawaii, and we can detect differences in the overall population level profile of the performance of the corals in these two locations. And what we're interested in understanding is how the environmental context is then influencing the biology.
And so do you have any preliminary ideas about
Yes, we
what's
do.
happening?
And it's actually rather surprising because we traditionally on reefs would say that a stable environment is the best thing for anything, right? But what we are finding is corals that live in parts of the reef where the temperature goes up and down fairly rapidly on a day-to-day basis, and there's quite a large range of temperature, seem to do better than corals that are in rather flat temperature profiles.
Do they do better out on the reef, or
Yep.
do they just perform better in your
in
chambers?
all settings. So on the reef, we believe they're doing better. And when we get hit by a disturbance that tests their performance thresholds, as is going to occur this year with the El Nino, we are able to test hypotheses that we can see how they perform here. We can now generate hypotheses that we can test back in the ecological setting, and that's exactly what we're doing. We have a parallel project that's going to be implemented here in Moorea, in Christmas Island, in the Line Islands, and in Hawaii, where we know that an El Nino will have very different impacts in those three settings, and we're going to be studying the same species of corals and how they perform in settings that are characterized by very high fluctuations and very low fluctuations. And we hope that through that analysis we'll be looking at it before, during the El Nino, and after the El Nino. So we are trying to characterize who will survive, what the context is for that survival, and which species are the most robust.
Exploring Our Fluid Earth is the dynamic curriculum developed by the University of Hawaii's Curriculum Research and Development Group. The award-winning Fluid Earth and Living Ocean textbooks are now interactive and online. New activities, updated content, and a teacher community. Exploring Our Fluid Earth is now freely available. Find out more at exploringourfluidearth.org. Welcome back. We're learning about the amazing physiology of reef-building corals, organisms that play host to algae living within their tissues.
You're looking at differences between individuals of the same species,
Yes.
but then also differences within populations.
So populations, so we go from the individual, comparing individuals within a population. We're looking at populations within a reef, and we're looking at different species on the same reef and on different reefs. So all levels of what we call taxonomy.
How do you tease apart, say, the better athlete, the higher performing coral, one individual from
Yes.
a population that is just outperforming another population?
So we are looking for all of those things. So we just want to understand why a given individual within the population is. So if we start with that question, we might go out onto the reef and we might say sample 20 individuals on the reef that are in a particular population. So 20 from population 1 and 20 from population 2. And maybe population 1 is in very highly variable conditions and population 2 is in very flat conditions. We'll take those corals and we expose them to identical stresses in the lab. So we essentially run them on the same treadmill. And we will be looking for the corals that do the best. And we can describe that by they continue to breathe relatively normally. Maybe they breathe a bit heavy to start with, but then they get into a rhythm and everything is fine. Compared to a coral that just starts to not look so good. It's starting to be very labored. And we've already talked about the fact that they have these small plants living inside them. These are these dinoflagellates or these unicellular algae inside their tissues. What's so great about those plants is they are actually responsible for the dark brown or green coloration of corals. And when corals aren't doing so well, they get expelled or lost.
And we call that bleaching, right?
We call that bleaching, exactly. So the colour of the visual colour changes and they become paler and paler and paler. And in fact, if they lose all their plants, they will die. And that's indicative of the fact that this is what we call an obligate symbiosis. It's absolutely required for the coral to survive. It would be like us losing our blood cells. You know, if we lose all our blood, that's an obligate symbiosis that we have with our blood. If we lose all our blood, we die. It's exactly the same thing with corals. And so, but what's great is that colour, or the changes in colour, is a very good indicator of how well a coral is actually performing. And so we can do these non-destructive visual surveys of corals that are in our experiments that are running on a treadmill. Some of them will be brown and they'll stay this fabulous colour all the way through the experiment. They're dark brown at the beginning and they're dark brown at the end. Other corals will be dark brown at the beginning, and then after a couple of days, they're starting to look a bit wishy-washy, a bit pale, and often we'll remove them from the experiment before they die. We're not interested in running them on the treadmill till they die. That's not our goal, because that wouldn't really be, you know, in forwarding our agenda. We're interested in the ones that are surviving, and we want to take the survivors. As I said, I'm an optimist, so I want to take the survivors, and I want to understand that biology of survival so well that I can first spot the survivors on the reef, that I can know who on the reef then I should be crossbreeding. And I'm so familiar with that in one species, and I understand how it works in so many species that I can immediately go to another species in a different location and say, you know what, in this population we want to look for the individuals that are found in setting A, B, and C. And that is going to be our source population. And by breeding those populations, we will have corals that are bred to withstand the future.
And the settings that you're talking about are like these areas of fluctuation on the reef where there's maybe more current, their temperature goes up and down.
And I always use this great example in Taiwan, because Taiwan is such an interesting place. I have a study site, it's called the National Museum of Marine Biology and Aquarium in the southern end of Taiwan. And they have a nuclear power plant that is inside a national park that was there before the national park was designated. And the effluent from the nuclear power plant is very warm water, and they pour it into the marine setting adjacent to the plant. And just off that nuclear power plant, there is the most beautiful coral reef that you've ever seen. It's flourishing. It's got almost 100% cover. Well, that is completely contradictory to our expectations. Now, what's fascinating about that particular setting is when we put our instruments in the water, what we were able to see is that it fluctuates in temperature actually, because there's a huge upwelling event that happens every day, and deep water comes to the surface at night. And so you have the hot water coming in from the nuclear power plant, but it's tempered by cold water coming in at night. So the corals are only exposed to this very warm water for a set period of time. And clearly the corals that live there now are used to that. It's so outrageous that they see 6 to 8 degree changes in temperature, centigrade, a day.
Wow.
A day. So these corals are more than super athletes. I mean, these corals, when I think originally the power plant went in, there probably was a large die-off in that area, as there was a selection. If essentially that experiment that I just described of running them on a treadmill, they will run on the treadmill until a whole bunch of them just collapsed and died. And what got left then were the super athletes. And because they were the only thing left in that natural setting, they were the only ones to breed together. And so now they have a population of extremely hardy corals. And it's, you know, many people are asking whether or not we should be using these very unusual circumstances where we know we have corals that are extremely well equipped to deal with the future, whether these should be reservoir populations for assisted migration. And what we mean by that is taking fragments of those corals and migrating them to a different location so we can essentially build the resilience or build for supercorals in a setting where they perhaps don't have any. So that is a discussion that is actually a current discussion. And again, it's fraught with problems. You know, that's sort of a biological intervention. And when we think about what our history with biological interventions is, it's not actually very good. And so it's that is much more of a, we can do the science, but that discussion is a discussion that has to happen with the communities and with the policymakers in the settings in which we work. And so, you know, I always say to people, you know, people say, well, where you're advocating doing something, why shouldn't we just be reducing CO2 in the atmosphere, for example, to sort of, to offset climate change? And I say, well, I can turn off my lights and drive an electric car, but past that, I can't. I'm a biologist. What I can do is I can explore the biology, I can understand it, and I can propose biological solutions. I can develop biological capacity, but the discussion as to whether we actually implement with that capacity is a discussion that's a much broader one, and is for somebody else to really take forward. But knowing that the science that we've brought to the table to inform this question is the very best possible, and certainly is at the cutting edge of what we're thinking in terms of ecological restoration as a field. And it is a discussion that we're having in many different ecosystems that are being very badly impacted by climate change. What do we do to ultimately do what we care about, which is to maintain the services that these ecosystems provide to humans? And, you know, we like to think it's about just doing it because it's the right thing to do, but they do serve our economy. And I think that really doing science that can actually render solutions that have economic impact is one thing that scientists generally try to stay back from that edge. Me, I just have stepped into that because I feel we can do something at this time, and it's our responsibility to do something.
Researchers and divers have reported extensive coral bleaching, including the loss of symbiotic algae across the Pacific as a result of the 2016 El Niño event that brings unusually warm water to the equatorial Pacific, stressing and often killing corals. The work of the Gates Lab may help these reefs to recover and adapt to our changing climate.
The Curriculum Research and Development Group in the College of Education at the University of Hawaii at Manoa. CRDG's training roots go back over 40 years. Through professional development programs, curriculum workshops, research on teaching methodology, individualized school and district training, and so much more. The Curriculum Research and Development Group, improving schools, improving education. CRDG.
Turn your love of the ocean into a lifelong career. Join NOAA, the National Oceanic and Atmospheric Administration, as we unlock the secrets in the deep oceans, track rapidly moving storms, model climate trends, protect and preserve our marine resources, and so much more. It's all in a day's work at NOAA. Find a career that makes a world of difference, enriching life through science, service, and stewardship. NOAA.
Healthy oceans are critical to our cultural, economic, and environmental sustainability in Hawaii. The ocean serves as a source of water, food, medicine, jobs, transportation, recreation, and energy. It controls climate and weather. Kosi Island Earth aims to share this ocean awareness by partnering with local scientists and educators to engage communities and schools in active science learning for an ocean-literate population. Kosi Island Earth is working to establish new avenues for connecting research scientists with educators and communities. Kosi Island Earth is enhancing the science and ocean literacy of our island residents and visitors. Kosi Island Earth is connecting scientific research, traditional knowledge, and ocean policy. Kosi Island Earth, bringing together university, government, research, and community partners to improve science education and ocean stewardship in Hawaii.